Let's talk about how we can use single-cell sequencing to help inform new potential strategies for immune monitoring and food allergy. In particular, I'd like to talk about two studies that we've conducted in collaboration with Wayne Trefler's group at Mass General Hospital to think about how do we track and identify the features of cells that are associated in the context of an intervention like oral immunotherapy, or in the natural progression or state of a disease like eosinophilic esophagitis. So we're going to look at two different strategies and how we can use single-cell sequencing to inform characteristics of the cells that are manifest in both of these two conditions, oral immunotherapy and EOE. For oral immunotherapy, the clinical study design is shown here. Starts with an enrollment in an oral food challenge to determine who can enroll into the study, followed by oral immunotherapy or small dosing by ingestion over a period of time with routine blood sampling collections at 4 time points. After a period of time, an oral food challenge is again administered to determine if tolerance has been induced, followed by a period of avoidance from peanut, and then a final food challenge to assess the durability in this time period of the oral immunotherapy. And from this, there are 4 potential outcomes. You can be fully tolerant through both of these 2 oral food challenges at the end and pass. You might have developed that you have the ability to pass the food challenge after the maintenance period, but not after the avoidance period, or you may fail entirely after the buildup and maintenance phase, as well as placebo groups who are participating. And so for this particular study, we have 12 patients, 3 in each of these different categories that we've looked at by single-cell sequencing. And the questions that we've been interested in asking in this study are related to what are the features of the peanut reactive T cells during the course of OIT. Some features of these cells might include the type of cell that they are based on phenotype, as well as the receptors that they express uniquely that identify them within their clonotypes. So the questions we're asking here are what types of peanut reactive T cells are present during treatment? How do these populations change during the course of oral immunotherapy, if at all? These cells, based on the sequencing data that we're going to obtain with their clinical status. In order to profile the peanut reactive T cells, we first take the blood draws that have been collected and we isolate the white blood cells. In that total population of peripheral blood mononuclear cells, we stimulate them with peanut extract for about 20 hours. This is a complex mixture of the antigens that are present in the peanut and allows us to assess a broad range of the functional responses elicited in the T cells in response to the antigen. Then those T cells that are CD4 positive and memory T cells are sorted out by flow cytometry and then used in a process for single-cell sequencing that we have developed over the last several years that's well attuned to small clinical samples. This process is called Seq-Well and is one that we've developed with Alex Shalick's lab here at MIT in the chemistry department. We've developed not only a process for isolating and characterizing the cells in their transcriptomes, but also in being able to enrich and recover in a matched fashion the TCR or the T cell receptors encoded by the alpha and beta chains that identify the clonotype. We have also developed a new strategy for the chemistries that are used that advance over the original ones that now allow us with high fidelity to capture the cytokines, transcription factors, and surface cell markers that are highly associated with the phenotypes of T cells that we know from immunology. Some of the original chemistries in this space for high throughput sequencing have not allowed for deep resolution of those factors, and the new chemistries that we've reported on recently in bioRxiv detail strategies for being able to uncover those pieces of information that are critical for being able to subset these populations. So now, in hand with the ability to identify deep profiles of the transcriptomes of single T cells as well as their TCRs, we have an ability to chronotypes and their phenotypic changes in time. In these data sets, in total for 12 patients, we have about 135,000 single T cell events that we have scored. You can see on the left-hand side of this slide the segregations of these based on surface expressed protein markers, canonical CD154 and 137 as activation markers, as well as a double negative population. So in the CD154 positive set, this is a group that we would associate with an antigen-dependent response in that CD4 population. You can see they segregate into a nice pink cloud. The CD137 population is one that would be more traditionally associated with regulatory type responses in humans, as well as the double negative. And so you can see, based on the protein expression, there is good segregation of these populations in the totality of the single cell data shown here. The segregation of these cells based on patient type is shown on the right-hand side, and you can see that there is a good distribution among each of these different subsets among the patients. Some patient-to-patient variability, but a high degree of overlap in these populations. I won't have time to detail all of the technical aspects of how we call some of these cells and identify them in here, but more than happy to share that information another time. One of the things that's really difficult about identifying T cells in RNA sequencing data from single cells is that the transcription factors and cytokines are often expressed at sort of lower levels and are noisy within their expression in single cell data. So to overcome some of these challenges, Brenda Munyon, former student in the lab now, developed a strategy for using an unbiased approach to asking which gene sets are co-associated with each other in these different data sets in a way that allows us to group them together into modules based on that covariation together. And you can see here in within the data set, we can uncover now modules based on this aggregate analysis to identify subsets of gene expression modules that make sense to us from an immunological perspective. In the top left-hand corner, you can see, for example, a Th2 biased module in the CD154, 137 double positive populations, as well as in a population of the CD154 positive set. And looking across the rest of these data, you can start to see the Th1 module, a Treg module that's highly associated with the 137 positive populations, as we would anticipate. And so in this way, we're uncovering known biology around these module sets, but allows us to identify the subsets of cells based on their Th characteristics. For each of the cells, or for the majority of the cells in those populations, we're also able to recover to a high degree the beta chain and the alpha chain from these subsets of cells. We can use the beta chain in this example to identify cells in the totality of the population that are clonally related to one another based on the CDR sequence that they express. And we can assign to those clonotypes the Th1, Th2, Th17-like signatures that we've uncovered through the unbiased analysis. You can see here that in the CD137 population and the CD154 population that there is indeed a expansion in the clonotypes and the clonal sizes represented in those groups, a good indication that we're recovering antigen-dependent T cells that are responding in a reactive manner to the peanut antigens. And on the right-hand side, you can see the types of gene modules that are associated with the CD154 positive cells based on the enrichment of those populations and their clonal size. So as clones get larger, you can see they also associate heavily with Th2 signatures. This is great. This is suggesting, in fact, we are recovering peanut reactive T cells and that they're expressing some of the gene signatures that we would typically expect to see in these populations in the context of food allergy. Taking each of those subtypes out based on the expression of those gene modules, we can then evaluate the Th1 subtype, Th2, and Th17 to further understand what distinguishes those populations in the transcriptomic space. And you can see each of those shown here in the individual UMAPs. There are 6 types that are most evident in these, and you can see they have characteristics that span from TFH1-like to TFH to TH-like. Within the Th2 populations, a TFH2-like population, Th2-reg, and Th2A, all of which have gene elements that are similar to reported phenotypes of these populations in other studies from mouse or from human. Furthermore, we can see that actually membership within these different subtypes is relatively exclusive based on the clonotypes. In the heat map shown at the bottom, you can see on the right-hand side the different subtypes and then the clonotypes that are represented. And you can see that based on the fraction of cells present in each of those, that they represent reasonably distinct clonotypic variants. some shared clonotypic identity between the TFH1 and TFH2-like populations, but the Th2A, as well as the Th17 and Th1, highly clonally distinct populations. One of the analyses that we wanted to do further on this particular question was to ask really, how do the TCRs relate to one another in those families? So looking explicitly now at the 6 types that we've just identified, we can ask, is there a high degree of relationship among the CDRs shown within the TCR sequences we've recovered? And so for 4 types, the TFH1-like, TFH2, TH1, and the TH17, we actually see that the overall similarity of the TCR types is very similar based on a metric like TCR-DISC developed by Paul Thomas's lab. And so what this really suggests to us is that there is perhaps a convergence of the TCR identities that's associated with these phenotypes that's driven by epitope recognition and a differentiation of the subsets. This is an interesting question, and one that we can't prove out entirely in a human model, but whether or not the epitope recognition helps drive or shape the overall response, or whether that's a memory effect based on prior exposure. Something that can't be resolved in these studies, but is an interesting immunological question about the development of these types of responses. Interestingly, though, we also have asked, how do these signature sets in the T cell populations that we see associate with the clinical outcomes of the patients that we've characterized here? And interestingly enough, through a combined method where we're looking at the modules of expression of genes within the different populations of cells, and then using principal components analysis to segregate those populations, we see in fact that there is a strong component in principal components analysis where we see a differentiation between partial tolerance, so individuals who passed at the maintenance stage but failed after avoidance, or the treatment failure group that did not pass either of the 2 later stage challenges, with Th1 and Th17-like signatures. In fact, some of these signature sets are present in the populations of cells from these subjects at the point of baseline, originally in when they first provided a blood sample into the study. This would suggest perhaps then that basal levels of inflammation are actually highly associated with the treatment failure. So to summarize the peanut reactive T cell story here, using single cell sequencing to uncover information about the CD4 antigen reactive T cells, I've shown you that there are 6 subsets of cells that we've identified in this particular study, and that the gene expression signatures of those are consistent with other data that are present in the literature on Th2A, TFH13, and Treg populations. This is good in that it supports and suggests that in fact other studies are seeing similar kinds of behaviors in the populations of cells during the course of OIT. Furthermore, the analysis that we've done here on those subsets using the clonotypic restrictions that we can infer from our recovery of the TCR sequences in these cell types show that there is a convergence on the TCR sequences that are used within these subsets. And it suggests that there's either a committed lineage that emerges from that specific TCR combination, or that there's an antigenic bias that has led to the amplification of those populations. Again, we can't resolve that with the data sets we have here, but I think it points towards an interesting relationship and uncovers further importance in relating the clonotypic identity of the cells that are identified with their phenotypic states, and perhaps tracking that information in time as well. And then finally, we've shown that the single cell gene modules that have been identified here in these cell types can be associated directly with clinical outcomes in this study, specifically with treatment failure. We've shown that there's a basal level of inflammation that can be a potential factor for determining or for assessing treatment failure outcomes. This obviously could provide extreme power in being able to think about enrolling patients into studies for oral immunotherapy or for prescribing such oral immunotherapies based on prior levels of inflammation within the individual. The signature sets that we've identified from single cells might be difficult to uncover through bulk methods, but potentially could be translated into other types of measurement tools that would allow for an easier evaluation of this, not requiring single-cell sequencing in the clinic. However, the technologies for single-cell sequencing are vastly and rapidly improving, and the ability to think about a future state where using single-cell data directly to provide for monitoring of patients is I think, a very plausible future state for immune monitoring. So I'd like to shift gears now in the last few minutes that we have and talk about how we can take a similar approach to uncover the populations of cells present in EoE, or eosinophilic esophagitis. In this particular study by Bert Reuter in Wayne's lab and Duncan Morgan, a graduate student in my lab, they've looked at biopsies from patients either with active EoE, in remission, or healthy individuals, and looked at biopsy sites from both the esophagus and the duodenum, and performed single-cell sequencing on the totality of cells that are recovered from these tissue biopsies. Again, the Seq-Well technology that we've developed with the Shalek lab is well suited for these very small populations and is easy to implement in a clinical lab setting, where we can take the cells that are disaggregated from the tissue and immediately load them onto these arrays for capturing the cell types with minimal other perturbations. Through the process of capturing those cells on the array, we can now identify by transcriptome the different populations shown here in the UMAP. You can see different populations that are present here. Particular, I'll draw a note to the teal-colored population, the small population that represents the eosinophils in this population. Obviously a hallmark of the disease, and nice to see these granulocytes captured in the single-cell sequencing data. You can also see there's populations of mast cells, and we've seen some evidence for neutrophils as well. There's a major population of T cells as well as some of the other epithelial, and what we can ask now in an atlasing sort of way is what types of cells there and what pathways are being expressed. Since we're very interested in T cell populations and the biology of those cells in relation to the disease state, we've emphasized the analysis that I'll share with you here on the T cells. So if we look now at the T cell diversity in the esophagus compared to the duodenum, we can see that there's a number of different subsets of cells that are present and shared in each of these different compartments, with one exception. So you can see there's a CD8 resident memory population that's similar in gene expression at the top here in the little circle map that's present in both the esophagus and the duodenum. Similarly, there's Th17 cells in both compartments as well as Tregs, but there is this particular subset that expresses a number of genes that have been reported and associated with the pathogenic effector Th2 population. And this population is only present in the esophagus. It's the yellow population in the top left of the esophagus UMAP. So this particular population is present in the esophagus of patients with disease. And in fact, what we see is that this population is statistically associated with active disease, as shown on the left in the fraction of T cells of this population E6. Interestingly, in remission, there's a higher population of the Th17 cells. In the center panel, we can also see the relationship between the fraction of the T cells in this pathogenic effector Th2 population in relation to the eosinophils based on immunohistopathology. So looking at the sample, we can now see there's a strong correlation between these two population groups. If we dig deeper into the gene sets that are enriched within this population of the P2Th2 cells, we actually see that there's a strong upregulation of genes that are associated with lipid metabolism, and in particular with prostaglandin synthesis. This is interesting because it suggests that actually these cells in the esophagus are expressing the pathways that are associated highly with prostaglandin D2 synthesis. Prostaglandin D2 is a chemoattractant that helps support and attract populations like neutrophils and other granulocytes. For the men in the audience, it's also associated with male pattern baldness. Interestingly, though, that means that there are a number of treatments that are in development and targeting similar kinds of pathways. This particular population, or this particular gene signature, can also be seen when we stimulate CRTH2 positive, CD161 positive T cells isolated from blood. So if we take peripheral mononuclear cells and we stimulate them ex vivo, and then perform single-cell sequencing, we can see that that population upregulates the same gene signatures associated with this enhanced fatty acid metabolism signature seen in the esophagus in the tissue sample directly. And so this actually provides a basis for thinking about a different kind of monitoring strategy for EOE patients. Instead of the standard of care with biopsy, it may be possible to think about strategies that would use a blood-based diagnostic to follow these populations of cells, either by frequency count, simply by the numbers of these cells, since they seem to have a high degree of active relationship with those in the tissue, or through kind of stimulated strategies like the one shown here. So just in summary of this portion for EOE, we've shown that pathogenic effector Th2-like cells are recruited to the esophagus during active EOE. They're present there but not in remission or in healthy subjects, and they're not present in the duodenum. They upregulate the genes that are associated with lipid metabolism that support the production of prostaglandin D2, and this seems to be a major acquired function of this particular population. Again, perhaps creating a positive feedback loop in the pathology of this disease that helps to recruit and maintain in the tissue granulocytic populations like the eosinophils. In addition, with thinking about translation of these data sets from the tissue into blood, we have shown that we can take CRH2 positive, CD161 positive memory CD4 T cells, and if stimulated ex vivo, those also then upregulate the synthesis pathways for PGD2. This altered lipid processing seems to be an aspect of that particular phenotype of the PETH2 cell type, and it may be a signature that we could use to improve monitoring strategies for patients with EOE or other eosinophilic diseases. So finally, I just finished and thank the folks that have been involved in a lot of the work here. I mentioned several of them as I've gone along. The Seq-Well technology and the improvements in the chemistries that are really necessary for enhancing the resolution of cytokines and transcription factors and other elements necessary for phenotyping T cells was developed by Todd Guerin with Travis Hughes and Mark Wadsworth in Alex Shalick's lab. The studies that I've shown here, the profiling of the T cells in oral immunotherapy was a very close collaboration between Brenda Munyon and Andy Tu, two former graduate students now in the lab, along with Bert Reuter and Wayne Truffler's group and Neil over in the Mass General Hospital. And the EOE study that I've talked about was spearheaded by Duncan Morgan in my group, and he's also contributed to a lot of the computational methods and analyses in the OIT studies as well. For those that are interested in Seq-Well as a technology, there's some information here. We've put all of this into a resource center at the Koch Institute at MIT. You can order chips and use them in your studies, and we have a number of online protocols now that describe how to use it, and we've transferred it to other labs that are interested in profiling clinical samples. With respect to funding, I'd absolutely thank the Food Allergy Science Initiative. This is a great program and one that's really been instrumental in us being able to bring some of these advanced technology tools into this area and apply them into the context of profiling food allergy. So thank you.